Hi, my name is Manish Gupta, and in this video, I'm going to continue discussing about AlphaFold, which is a model for accurate protein structure prediction. Okay. In the previous video, I talked about what is AlphaFold, why it is important. Basically, it's a model to be able to find the 3D shape from the genetic sequence, right? From the genetic sequence of various proteins. We also observed that AlphaFold is very accurate. So in fact, in CASP 14, in this challenge, essentially AlphaFold led to the best results. Lower is better here. So it led to the best C alpha root mean squared deviation, right? It can actually accurately predict the 3D structure of proteins, even very long proteins, very long protein chains. Okay. Then we talked about, yes, it is not just accurate, it's also reliable. It can actually come up with a confidence score using which users can actually decide whether they want to consider the 3D structure predicted by AlphaFold or not. AlphaFold mainly takes the input amino acid protein sequence as input, right? And these proteins, basically, when the amino acids are a part of those proteins, they're called as residues, right? And then it basically tries to search for similar proteins and similar sequences and uses them as extra input. So they are obtained by doing a genetic database search so as to obtain multiple sequence alignments, or by doing a structured database search so as to obtain these pair representations. Both of these are processed quite some using an input network so as to essentially get a MSA representation and a pair representation. And then AlphaFold basically comprises of two main blocks, evoformer and structure module. And as you can see, the evoformer guy essentially runs multiple times. So there is multiple times to essentially recycle this in that senses or do iterative refinement. And you actually also do iterative refinement after the structure block, right? So in the previous video, I talked mainly about evoformer. We went into deep details of evoformer and also the details about the inputs that are passed to AlphaFold. So AlphaFold takes, as I mentioned, three main inputs: the input sequence, MSAs, and paired representations. And then it basically has like features associated with each of them. So that's what we discussed in the previous video. We also talked about detailed evoformer architecture. Essentially, it is a two-tower architecture, one to process the MSA representations, the other to process pair representations. And we discussed details about how each of these blocks in the evoformer architecture work. So we first looked at the row-wise gated self-attention with the pair bias, We also looked at the column-wise gated self-attention here, right? And then we looked at the transition block. Then lastly, we looked at the outer product mean block in the evoformer super block. We then also looked at the tower for the pair representations, and then we talked about triangular multiplicative update and then triangular self-attention network, self-attention modules, right? So that's that. This is where we stopped last time. Let's basically continue from there. So what is the architecture of the structure module? So now that we know the architecture of let me get back to the picture. So of the evoformer module, next in this video, I'm going to talk about how does structure module work and how does it really come up with this 3D structure of the protein molecule. Okay. The structure module looks like this. It basically takes a single representation of size R cross C from the evoformer block. Remember, the evoformer block basically gives out the MSC representations and pair representations. So the pair representations essentially is coming up there, and the single representation is there. So the single representation is a representation obtained from the MSA matrix, MSA tensor, essentially. Yeah. Okay. So evoformer provides essentially a highly processed MSA and a pair representation. So the single representation is basically the first row. It's basically the first row and its projection. So you take the linear projection of the first row of the MSA representation and you get the single representation. That's that. Okay. You also, of course, have the pair representation anyway. And what the entire structure module does is to map this single representation and the pair representation combined to concrete 3D atom coordinates. So that's basically the entire molecule is basically what is obtained. The shape is what is obtained as the output of the of this structure module, right? So it comprises of 8 blocks and they have shared weights. So, and each layer basically updates the abstract single representation. This representation, it updates and comes up with this revised single representation, as you see here. Okay, it also comes up with the what is called as the residue gas. It's called a residue gas, but basically it's the coordinates of the backbone frames and the orientation of the backbone frames, how they are oriented and where they are, right? So compared to the origin. So that's that. Of course, at the end of it, basically the the last block is basically going to give you the overall, overall 3D representation of the entire molecule. Okay, so this residue gas is very interesting. So basically, it basically comprises backbone frames, as I mentioned, the position of the backbone frames as well as the orientation of those backbone frames. Okay, so essentially, the output from the structure module is in multiple parts. One, it basically tells you, a protein molecule basically comprises of a backbone and side chains, right? So backbone is basically N, C alpha, and C kind of a triad. So it basically this backbone frames tell you how many of those N, C alpha, C are there in the entire molecule and where they are placed. So of course, how many are there is already known in the amino acid sequence, but where they are placed. So what is the position of each of those, as well as the orientation. So the dihedral angles, the phi and psi, are also predicted basically here, right? While the dihedral angles of the side chains are basically what are predicted at the top from the last layer in that sense, or rather from every layer, but then the last layer ones are the ones which are used, which are finally outputted. Okay. So that's that. So essentially, residue gas is encoded in the form of what is called as TI, and it's basically one backbone frame per residue. So if you basically had like 256 residues, for each of them, you would basically have like a backbone frame, which is predicted, TI, which is predicted here. And as I mentioned, TI basically comprises of two things: rotation and transformation or translation. And translation basically just tells you how translated from the origin is this backbone, and how it is rotated. So basically, rotation is a 3x3 matrix, and the TI translation is basically just a bias. So just three numbers, basically XYZ, three numbers. Okay. Now, of course, the single representation and pair representation comes from the Evo former. Where does this backbone frames come from? So in the beginning, you basically just initialize these backbone frames such that all of those backbones are at the origin. And essentially, the 3x3 matrix is just an identity matrix. So they're all oriented in the same way. Okay, so that's called black hole initialization. So all residues are located at the origin of the frame with the same orientation. Okay, and then over iterations, essentially, you keep there is an IPM module which basically keeps predicting how much you should rotate and how much you should translate these backbone frames so as to reach a particular, so as to come up with a revised representation of these backbone frames. Okay, and the revised representation is basically which is shown here. So each of these basically predict TI, and then the TI is sort of applied on the current local representations, so as to essentially obtain a current local coordinates, so as to obtain the global coordinates in this way. So TI is applied to that. TI basically comprises of Ri and small ti. So small ti is basically for a translation, but this Ri, since it's a matrix and X local is a vector, it basically rotates the vector. So, and therefore comes up with a 3D orientation. Okay. So now how are the atom positions in 3D structure obtained? So, so essentially each residue here essentially is like it consists of a rigid frame, as it is called. So in the sense that if you think about it, the N, C alpha and C are already known. So they're basically, the bond lengths are known. So you don't really vary the bond lengths. So you know that N and C are going to be connected to this bond length, and N C alpha is basically this bond length. So bond lengths and bond angles are actually known. Okay. So what is unknown is how much torsion, how much 3D rotation do you have in each residue. Okay. So basically, therefore, in these backbone frames and also in the predicted chi angles here for the side chains, what is not fixed is basically the torsion angles, the rotation, the 3D rotation angles. The bond angles and the bond lengths are always fixed. So they're basically known, and they are fixed even in typical chemistry, typical molecules. They're just fixed. So therefore, you don't really mess around with them. The only thing that is variable is how much do you rotate 3D in a 3D manner so as to basically obtain a particular kind of a protein molecule. Okay. So that's what is supposed to be predicted in that sense. And the way this is predicted is by defining a rigid group for every of the 3 backbone and 4 side chain torsion angles. Okay, we'll talk about what is this rigid group, but there are like overall 7 different torsion angles to be predicted. Omega, psi, phi, and then chi1, chi2, chi3, chi4. Omega psi phi are the dihedral are the angles for the backbones themselves, while chi1, chi2, chi3, chi4 are for side chains. Okay. Now, we already talked about phi and psi, which is basically the angle between C alpha and C alpha C, those bonds. But then there is also this redundant angle in some ways, omega, which is basically between N and C. Now, of course, if you have fixed phi and psi, your omega is sort of fixed, but it's good to predict all of them and then apply violation constraints as to check if they are not violating and making sense in that senses. Okay. So they define rigid groups. Now, what do rigid groups mean? Rigid groups basically mean that if I basically, and in this table, what you see is 20 amino acids, right? So they are representations for each of those 20 amino acids, okay? So here is the backbone. So backbone is NC alpha C. Most of the cases, most of the cases it's NC alpha C and C beta, but only in glycine, it's basically NC alpha C, okay? And then there are other atoms. So what you see here are other atoms, and the positions of these atoms basically depend on the previous ones. So in the sense that the position of oxygen atom is going to depend on NC alpha C, C beta, and this angle called phi. Okay. Similarly, in this one, the position of the C gamma atom is going to depend on the chi1 torsion. The position of C delta is going to depend chi1 as well as on phi. Okay. So position of C delta in ARG amino acid is going to depend on chi2, chi1, and phi. Okay, so that is basically how they've defined rigid groups. So basically saying, that predicting the position of N, this nitrogen atom, is very difficult because it depends on the predictions, appropriate predictions of, the backbone plus phi plus chi1 plus chi2 plus chi3 plus chi4. So it's basically complicated to predict those which are towards the right side and more easy to predict the positions of those which are on the left side. Okay. So now that's that. So basically our structure module comprises mainly of this IPA module, right? After that, there is this predicting relative rotations and translations and predicting chi angles, but they are sort of predicted using some dense layers. That's all. I mean, there's not much to it. Or sometimes convolution layers. But the main part is in this IPA module. What does this IPA guy do? What is this IPA guy anyway? Right? So the IPA guy stands for invariant point attention. Okay. So it's basically going to take these three things as input. It's going to take the pair representation, it's going to take the single representation, and it's going to take the current backbone frames as input. Okay. Now, and remember these backbone frames, what is the size of each of these? So pair representations, the size is RRCZ, which is basically for every residue, for every pair of residues, there is a CZ sized feature vector. Right? Single representation is RCM, which is basically for every residue, you basically have a CM sized feature vector. Backbone representation is RC3 cross 3 and R3. So for every residue, you basically know the translation coordinate from the origin and the rotation, basically how it is rotated from the typical 3D frame, okay? Standard 3D frame. Okay. So essentially, what does invariant point attention do? Well, it basically just comes up with an updated single representation, considering the pair representations and the backbone frames. So considering, where your current backbones are, you may want to come up with a better representation of each of those residues. That's basically what IPA does. Okay. So it basically comprised of 3 different pipelines, as you can see: the blue pipeline, the red pipeline, and the green pipeline. Okay. So the red pipeline is the easiest to understand. It's basically just standard self-attention. There is absolutely no difference compared to standard self-attention. It's basically just a sequence of R tokens, each of dimension Cm, and you just do standard QKV transpose, right? QK transpose, softmax, and then multiply by V. So it's a standard multi-hit self-attention. Of course, there are, H is head and C is channels here, right? So H is head, C is channels, and you basically get the revised representation. So then, the blue and the green guys, essentially those pipelines try to come up with a revised and updated single representation by considering inputs from the pair representation and the backbone representation respectively. Okay, so the pair representation guy interacts with the with this representation of this with the single representation in the same way as our outer product mean guy interacted. Okay, so the idea is that when you're computing QK transpose, you already have this pairwise interaction matrix in some ways, residue pairwise interaction matrix, and you basically just add that pairwise matrix out there. Okay. Of course, you can do a little bit more. So what you could also do is basically take the same paired representation and use it so as to also impact a gating kind of a thing. So essentially, what you could do is basically use this to define how much importance this residue should pay to other residues in the sequence, and that is also controlled here in the blue pipeline. Okay. Now the green pipeline basically, yeah, it's basically R3 cross 3 and R3. So what you do is to, of course, use dense. You first basically take these local coordinates and convert them to global coordinates. And the way you would do that is, of course, you take the, the keys and the queries and the values from here, and you interact those keys and values from here with the local coordinates so as to basically obtain the global coordinates. Or the coordinates in the global frame in that sense. Okay. So once you do that, you make them as a part of those. Of course, once you have those queries and keys, along with interactions, local coordinate interactions from the backbone frames, you essentially already have a squared matrix, which you can again add to this dot product. Okay. Similarly, you can take the values from here and then again create a squared attention matrix and have it also impact the overall final computation that you have for your updated single representation. Okay. So that's basically, it's an invariant point attention as it is called. Essentially, it doesn't make use of information from other MSAs because it just makes use of the single representation, therefore point attention. But at the same time, it basically makes use of templates via pair representation. Okay. So now that we know this entire structure of the AlphaFold network, how is it trained, right? That's the other important part. So it is trained in two stages. The first stage is called pre-training. The other is called fine-tuning. So of course, pre-training does not make use of any 3D structure. Fine-tuning makes use of 3D structure in that senses. Okay. Both of them make use of 3D structure, but in different forms. So fine-tuning actually has more losses. So essentially this experimentally resolved loss and violation loss, while pre-training actually has just these 5 losses. Okay. Let me talk about these losses one by one. So of course, overall, you see there are 7 losses. So I'll talk about some of them here and some of them on the next slide. Okay. Let's start with the FAPE loss or the frame aligned point error loss. So here the idea is you take the predicted atom positions and you take the true positions and you compare them and design this frame aligned point error loss. It's basically like a little Euclidean distance kind of comparison, but it is done under many different alignments. Now the idea is if you look at a molecule this way or you look at a molecule in a slightly different way, you can basically see that the distances differ, So therefore, what people do is to essentially, compute these FAPE aligned point error under different alignments, and then they take the averaged out loss, averaged out distance, differences in distances between those main atoms. Okay. Now, auxiliary loss. So auxiliary loss is basically evaluated from the structure module at every iteration. So essentially, it's not like you just computed the last one. You actually compute this loss at every iteration. So it is averaged FAPE loss and torsion losses on intermediate structures. So whenever you're basically doing these iterations several times, you basically get intermediate structures out. You compute the auxiliary loss, the FAPE loss, and the torsion losses. So the torsion loss is basically computed as the cosine of the angle between the predicted and the true torsion angles. So you have all of those torsion angles, and phi, psi, chi1, chi2, chi3, chi4 for each of them. You actually compute take the cos and then take the differences. Okay. There's this dist loss. So dist loss is basically the distogram-based loss. So distogram is distance histogram in that senses. Okay. So the idea is that you essentially take the C beta atoms across each of those residues, and you take the distance between the C beta atoms of each pair of residues in the molecule, and you essentially, you essentially compute a distance histogram, the predicted distance histogram. What is the predicted distance histogram? You take these distances of between every residue pairs defined as the distance between their C beta items, right? And then you group them into 38 bins, right? So these 38 bins basically, so you have a number of pairs lying in each of those 38 bins, and you have number of pairs lying in each of those 38 bins from the actual true molecule, right? So you have a predicted distribution and you have a true distribution, also called as predicted histogram and true histogram. You compute cross entropy loss over those. Okay. Then the MSA loss. MSA loss is an average cross entropy loss for masked MSA prediction. So remember, we did some sort of masking when we were coming up with MSA representations. So how does the masking work? The masking works just like in BERT, very similar. So you mask out 15% of those residues in MSAs. Where out of those 15%, 10% are replaced with another amino acid, 10% are replaced with amino acid with an amino acid sampled from the MSA profile for the given position. So the idea is that you basically have several MSAs, and then you look at that position, and then you basically sample from given that weighting, given that given what are the popular amino acids in that position. 10% are 10% you don't replace the amino acids at all. You just keep it as it is, and 70% amino acids are replaced with a special mask token. Okay, so essentially there's a mask token. There is a no replacement. There is a replacement with a global any random amino acid, and there's a replacement which is locally sensitive. So to that position, I mean, in that position, what most frequently occurs, you sample from that distribution. Okay. So overall, when you're trying to predict the MSA position, the right MSA at that position, it could be any of those 20 amino acids are known gap masks. So basically, therefore, it's a 23 class cross entropy loss that you define. Of course, the idea is that the network should be able to predict the MSA at the end, right? By the way, these losses are essentially defined either by doing these losses are typically defined right over the Evo former last output. You essentially apply dense layer so as to basically define these losses. Many of these heads that you see here. Okay. Confidence is the model confidence loss. So basically, they project the final single representation, the single representation that you obtain from the Evo former block. From the rather single representation that you obtain from the structure block. Well, the structure block also gives you a single representation, and you basically map it to 50 bins. And the idea is that it should be able to predict this thing called as the IDDT C alpha, a metric that you can compute from true molecules. Okay. So the idea is that this head is also trained as part of training. And then the nice point is, given a test sample, you can actually use this network so as to predict across 50 bins what is the confidence level about the correctness of your 3D structure prediction. So that's that. There is experimentally resolved loss. Now, what is this? So basically, there is a head that you apply on top of the single representation, just a single neuron, which tries to predict if an atom is experimentally resolved or not. So experimentally resolved basically means that the residue, the ith residue was determined with a certain level of precision. Okay, so the position is determined with a certain level of precision. You basically call it technically as experimentally resolved, and all that this guy does is to do a binary classification predicting whether this residue was experimentally resolved or not. Okay. Lastly, there is this violation loss. So when you come up with structures, now structures may just violate physical constraints, right? So basically, of course, the bond lengths and bond angles for the C alpha and for the backbone were fixed, but then you see the bond lengths, bond angles for the side chains are not fixed, and they basically need to be predicted, right? Now there could be some incorrect predictions that may happen, and therefore they basically come up with three violation losses. So one on the bond length, the second on the bond angle, and third on the atom clash, right? So I mean, you basically take two non-bonded atoms and you try to see what is the distance that is predicted, distance in the predicted structure versus the distance in the actual structure or in the literature, right? And you check if they are at least at least this margin away or not. If they're not that margin away, then you basically count that towards the loss. So basically saying that, hey, they're too close. They're not supposed to be so close to each other in that sense. Okay. So that's that. So that's basically overall architecture of AlphaFold. Now, of course, AlphaFold has many evoformer blocks. How many evoformer blocks and MSAs are required really, right? So basically, when you do this similarity search, you also obtain multiple similar sequences. How many of them are really required, right? So they did this experimentation, and they found that evoformer blocks overall, you see, there were like 48 blocks, but then they also repeat. There is iterations also going on. So they found that if you look at the structures which are obtained from these each of those blocks, you can actually try to compute some metric from those structures. What they observed is that across three different proteins, they observed that yes, some proteins basically get predicted much earlier in the entire iterations, but there are some proteins like this green protein which really requires the entire length of the network, all the 192 evoformer blocks, so as to be able to predict correctly. Okay, that's that. Now, on the right side, what you see is how many MSAs are required. So basically, by the way, on the left side, the higher the value, the better. On the right side, essentially, this is also higher the value, the better here. Okay, and on the x-axis, what you see is the median per residue n for the chains, in the sense how many MSAs are you considering in that census. Okay, how many MSAs you're considering. And as you can observe, if you consider more and more MSAs, typically you see better accuracy, right? Typically that leads to better accuracy. However, after, let's say, some MSAs, after say 100 sequences, this sort of saturates, right? After 100, there is not too much of worth. Remember, the more MSAs that you take, the more the compute power that you require, the more latency it will incur and so on, right? So, of course, you want to really reduce the total number of similar sequences that you consider. Okay. But the interesting part is that, yes, I mean, just with about 100 sequences, you more or less are able to obtain all that you need. I mean, most of the accuracy that can be obtained using up to 100 MSAs. Okay. So how about the latency of this AlphaFold model? Well, I mean, they proposed an ensemble-based model of five different AlphaFold models, but then they observed that without ensembling also does not really lead to much drop in accuracy. So without ensembling on a typical V100, it takes about 0.6 minutes to process a protein with 256 residues, 1.1 minutes with 384 residues, and 2.1 hours with 2,500 residues. So, if you really think about it, for every protein, it still takes about 2.1 hours if you basically look at longer proteins. But hey, this is much, much smaller compared to years that the typical non-deep learning methods take to basically predict the 3D structure of proteins. Okay, so across these two videos, I tried to capture the details of how AlphaFold works. AlphaFold is such an interesting architecture, right? I think it basically revolutionized the field of drug discovery, or even basically, computing the 3D structure of proteins for other kinds of tasks, right? So broadly, the architecture is very interesting. Of course, it takes input as the amino acid sequence of the current protein, which you want to figure out the 3D structure for. It takes the MSAs, the multiple sequence alignments. It also takes templates, so pairwise representations, of interactions between those residues, right? Based on various other related, various other similar protein sequences, right? It outputs three things. It outputs atom coordinates. Where are those 3D atoms or atoms in the 3D coordinates? The histogram, it basically also gives you the distance between those, right? And also per residue confidence scores. It also tells you, right, hey, these are the confidence scores for each of those residues. So that helps you, that helps anyone who is basically analyzing the structure to take this structure with a pinch of salt, this predicted structure with a pinch of salt. If the confidence for a particular residue is low, they may want to basically just try and focus on that part of the molecules as to understand the structure better using standard methods. Okay. So, and overall, there is a lot of novelty across the entire architecture. It's an iterative architecture with two blocks, the evoformer block and the structure block. In general, these two blocks try to jointly embed the MSAs and the pairwise features. They come up with a new output representation and an associated loss that enable accurate end-to-end structure prediction. Right? They come up with a new attention architecture in that sense, several different forms of attentions, some of them trying to accommodate for the physical violation constraints, some of them trying to just come up with revised representations of those MSAs and therefore the original amino acid sequence as well. There are intermediate losses which are also used to achieve iterative refinement nicely. There is masked MSA loss, borrowed from the BERT literature, from the transformers literature broadly, right, to jointly train with the structure. There's also learning from unlabeled protein sequences using self-distillation. So remember, they basically trained their model in two stages. First stage, they basically just trained the full model, and then they basically applied on unlabeled data, obtained labels for them, high confidence ones they used to do second round of actually training the model. So two rounds of self training or self-distillation, as you may call, right? And then the nice point is that yes, the overall architecture gives you self-estimates of accuracy in terms of per residue confidence scores. So an interesting model. Yeah, fun reading about it. Hope you like the video. So, that's it. Thank you for watching. Connect with me on my LinkedIn or look at my research on my homepage. Thank you.